we continue with this agenda and this cutting-edge innovation from the i4KIDS ecosystem research groups, 4 groups, more than 30 entities, universities, hospitals, and research centers with a portfolio of more than 90 research projects focused on foster and promote pediatric and maternal health innovation. Takes part of this. These snapshots are a glimpse in the cutting-edge work happening across the i4KIDS ecosystem. So imagine, from novel diagnostic tools, like the ones, whoa, oh no, what our foundation, the vase, no, la piña, no caigui. Imagine, let's continue with this idea. From novel diagnostic tools to therapeutic advances, each project that's going to be presented in this stage represents the future of pediatric healthcare. First, pediatric healthcare faces unique challenges that require solutions specifically tailored to children's needs, from diagnostics to treatment and beyond. By supporting these innovations, we're not just enhancing pediatric healthcare, we're investing in the future well-being of entire generations. So that's why it's so important to give us space to give all this support to this kind of projects. And today we are here to tell the world we need more commitment, we need more invest. So what we are going to hear is 4 bites of 4 big projects, small. It seems to be small, but it's a big project, like this one. It seems to be not fragile, but it's yeah, strong enough. 4 big projects that would deserve a whole day in a stage. It's my opinion, but we are lucky to have 4 lines of research that go further to offer great solutions to concrete realities. We want to we want to present. So we are going to start with the first group, Muscle on a Chip for Duchenne muscular dystrophy, with Javier Ramón Azcón, group leader of Biosensors for Bioengineering, Institute for Bioengineering of Catalonia, IBEC. Yes, hello, nice to meet you. Javier Ramón Azcón is a group leader at the Institute of Bioengineering of Catalonia, IBEC in Barcelona, Spain, where he leads a Biosensors for Bioengineering group. And his research integrates sensor technology and nanotechnology with tissue engineering to develop organs on a chip. OOC, yeah? Javier, wait, yes, we are all learning step by step. Javier has established significant national and international collaboration and also has entrepreneurial experience. So the stage is yours, and you have 10 minutes. Well, nice. No time? Yeah, I think you are trying to do your best. Okay, thank you very much, Javier Ramón Azcón. Here you are.
Okay, yeah, perfect. So, well, first, thank you very much for the invitation to stay here. It's a pleasure, and it's really nice to have all these people working in different areas of research, entrepreneurs, investors, funding. Our work in our group is focused in organs on a chip, okay? Organs on a chip, probably you know already, but it's basically to try to minimize or miniaturize one function of that one tissue, okay, and put it together in a microfluidic platform in order to mimic one phenotype that's interesting or one specific function of the tissue that you want to test. So we are not trying to make a full liver, we are not trying to make a full kidney or whatever, because this is not able at the moment, it's not possible. But what we are interested in is to try to get this small functionality in an in vitro system. Well, you know already that the drug development is a long process, it's very expensive, so organs on a chip can help a little bit in these preclinical phases, because we can get better data, okay, we can provide better data to the clinicians, to the drug pharmaceuticals companies, and also we can make reduce the cost, okay, of these preclinical phases, and reduce the animal experimentation. That is, if possible, is also nice. We are in the preclinical phases. We can provide different kind of organs on a chip to different approaches. In our group, we are more focused in the first step in diseases on a chip models. And basically, what we are trying to do in our group is working in these 4 tissues. We are working in pancreas, we are working in liver, adipose, and skeletal muscle. But not only in these tissues, okay, separately, but how they interact within them, okay? So how healthy pancreas, for example, can affect liver with NAFLD, or vice versa, no? Have an NAFLD model in liver or another disease can affect a skeletal muscle or adipose tissue. To do that, as you can imagine, it's not easy, so it's quite complicated. We have different areas of research in our group that are focused in different parts. We have people working in biomaterials, people working in tissue engineering, in sensors, in microfabrication, and we try to synthesize and put everything together to get our organs on a chip. Today, we will focus in this disease, that is, we have developed a model. So we are working in Duchenne, a muscular dystrophy model. Basically, this disease is a rare disease, even it's a pediatric rare disease, but even it's a rare disease, it's one of the most prevalent disease in the pediatric. It's a severe progressive disease that affects the children from very early stage. And basically, now at the moment, it's not cured, okay? It's only palliative, we can say palliative ways to improve the quality of life of the children, but it's not possible to cure them. So our work in this disease was to try to mimic this disease in vitro, so try to make a model, a human model of this disease, and use it as a way to study the disease, but also to test different drugs. So the basic problem of this disease is that it's a genetic mutation and the children cannot produce dystrophin. Dystrophin is this protein that connects the actin system of the skeletal muscle to the basal lamina outside through the sarcolemma. No? So these patients, these children have no dystrophin, so when there's a muscle contraction, the sarcolemma suffers a lot of damage, okay? And one of the things that happens, for example, that liberates creatine kinase. Of course, the muscle can regenerate, okay, because it's an organ that can regenerate a lot. But when this happens a lot of times, well, basically the muscle is replaced by fibrotic tissue and fatty tissue. So they are losing muscle in the early stage of the life. Well, so our idea was, okay, let's try to do this model. Let's try to mimic this model in vitro. So our first idea was, okay, let's try to make a three-dimensional model, functional model of this muscle, okay, to try to mimic these phenotypes. So basically, the thing is that the three-dimensional is very important because all the organs are in 3 dimensions and not in 2D. And this 3-dimension makes the functionality of the tissue. So if you don't have the correct shape, you don't have the cells organized in a right way, they are not functional, okay? They cannot reproduce the functionality of the tissue. In this case, the contraction of the tissue. So in our lab, we have different approaches to fabricate 3-dimensional tissues. We have 3D bioprinting, electrospinning, molding. In this case, we choose molding because it was easy to produce in, we can say, no high-throughput manner, but in a high-scale manner, the tissue. So we developed these molds using just 3D printing, where we can produce these small chips, okay, with 2 pillars, okay? In these 2 pillars, in these small chips that looks like a pool, we can introduce the cells together with a matrix of biomaterial, and we can grow the cells inside, and the cells stay in these 2 pillars, and at the end we can electrically stimulate and check the movement, the contraction of the tissue. So the cells are from patients. We are working with different cell lines that are provided with Benedict Chassot from the group, from French group, working in this disease also. We have like 9 different cell lines from different patients and 7 cells from healthy people. We developed these muscle tissues. I told you this is the protocol for developing the tissue. They look like that. So at the beginning, it's full of the biomaterial, the tissue. In day 0, the material contract through the pillars that we have in the system, and it looks like this in the plate, as you can see. And we check how the maturation of the tissue, because they are highly aligned, they are highly mature, and when we apply electric stimulus, but then it contracts. Okay. So this is very nice because we can, well, the tissue looks like this. This is a full imaging of the tissue, okay? It's very small, as you can see. It's nice because we can make histological analysis. As you can imagine, we can cut, we can make cross-sections and check the different proteins and how they are organized inside the tissue. We can check for dystrophy, for example, that is the main problem of this disease. But also the important thing is that we can make functional analysis. This will be required, okay. So we can apply electrical stimulus and we can record the movement of the tissue, and with that we can analyze the fatigue of the tissue, the force of the tissue, and how is the behavior of this skeletal muscle. So we can apply this, we can say low frequency, stimulus, like a twitch contraction, okay. I wait until the video finishes because if not it will crash for sure. Okay, now. And we can apply also the more higher frequencies and to check tetanic contraction. No? That is this contraction that it requires sometimes to relax, okay, and the tissue to become relaxed again, okay? Yeah, perfect. So with all this information, then we study the phenotype of the disease. So we make a cutting, we cut the tissue in different pieces, and the first thing we were observing is how the dystrophy we have dystrophy or not. Then the first phenotype was clear. We don't have dystrophy on the patients, okay? That is what we were expecting. The second phenotype was, okay, what happens with the tissue when it's contracting? And our surprise at the beginning, a small surprise, was, okay, the healthy tissue and the tissue from the patients looks the same. They have the same force. Oops, sorry, I make a spoiler. Okay, it's the same force, they behave the same, but if you think about that, it is normal because these these kids that have this disease, the muscle is okay. So the muscle is healthy, that they can contract. The problem is when they contract after the exercise. So when they start to have exercise is when the muscle receives damage. So we make this, so we make a regime of stimulation to the tissue, so we simulate the exercise on our tissues, and the phenotype was clear. So after exercise, the tissue starts to lose strength and to start to have fatigue, okay? So we can show this tetanic fatigue-like behavior in our tissues. And the functionality of these tissues allow the induction of this damage after the exercise. We validate this damage using Evans blue. Basically, Evans blue is a dye that only enters in the skeletal muscle when it suffers damage, when the sarcolemma of the muscle is damaged. So we demonstrate in this example that the tissue from patient from TMD, the Evans blue enter inside the myotubes. Okay, so to take profit of that, we integrate in this organs-on-a-chip system some kind of biosensors. I will not enter in detail, but basically are plasmonic biosensors that allow us to measure in real time some proteins are secreted by the tissues. Okay, so it looks like that. The prototype, we have this big platform where we have the tissues, in this case 6 tissues, connected by a microfluidic system, and connected to our LSPR sensors that allow us to monitor the creatine kinase that is only liberated when the skeletal muscle is damaged in real time. So we start to make exercise to the tissues, and then we monitor the creatine kinase that was liberated when the tissue was damaged. We validated that by ELISA. This is how looks the microfluidic system, okay? With the different sensors. This is the 1 minute, but well, I will go fast. This is how they correlate, no? The ELISA with the biosensor, okay? And this is how looks the final platform, no? We have the organ-on-a-chip with the tissues, then the sensors that allow us to monitor the level of creatine kinase. And this is just an example of how it looks, no? When we start the stimulus, we observe the liberation of creatine kinase from the skeletal muscle. So we take profit of that. I will skip that, okay? But basically, we contact with one company in Barcelona, a biotech company that give us different drugs to test. We tested them in our model, and basically, we demonstrate that one of them, the number 13, that is confidential, was the one that saw better results in fatigue, but also in upregulation of the utrophin, that is one protein related with dystrophin. Now we are working with 2 more companies, one from China, that is called Ratingen, that they give us the drugs, and we are testing them in our model. And we are waiting for another collaboration with what biotech from Canada that also they want to test some drugs in our model to, well, to be used in this disease, in Duchenne. So, well, this is how looks the final device and the take-home message that we developed this system to test or to have the phenotype for Duchenne. We are able to evaluate the effect of different drugs on them. And in principle, this is a tool that can be used for preclinical research. This is our group in IBEC. Okay. And this is the funding and collaborators. Thank you very much for your attention.
Yes, you got it. Thank you very much. Javier Ramón with this muscular chip for Duchenne muscular dystrophy. This first research. So next one, group 2. This practical application of pharmacogenetic biomarkers in child and adolescent mental health disorders with Dr. Amaia Hervas, head of Child and Adolescent Mental Health Service at the Hospital Universitario Mútua Terrassa. Dr. Amaia Hervas, MD, and member of the Royal College of Psychiatrists, PhD, head of Child and Adolescent Mental Health at the hospital, as I said before, in Terrassa, and co-founder of IGAIN, principal investor in projects are autism and ADHD, with more than 150 publications and 300 presentations. Indeed, she's also coordinator of a master in child and adolescent psychiatry and international trainer in AC diagnosis. Member of scientific field. So thank you very much, Amaia. So it's your turn. The stage is yours.
Well,
10 minutes. That's
yeah.
Now.
Thank you very much for inviting us to present our work. This is the work of a team working in University Hospital Mútua Terrassa. And we are a research group. We are an adult research group in autism. And also we are a Texan group, and we are part of several genetic consortium. And we have several areas of research. We work in etiological, especially in neurodevelopmental disorders, genetic factors. And also we are implementation, artificial intelligence, virtual reality biomarkers for clinical measures. At the moment, we have an horizon that we are coordinating the case 3, and we are using artificial intelligence for prediction of suicide in adult children and adolescents. Suicide is very much increased in autism, especially normal, acute, and verbal functional patients. We also have a RETOS grant that we are just finishing, and we have developed a virtual reality simulation program for social cognition for children between 6 and 9 with biomarkers, and that I think for us has been very important because we are not seeing any relationship between high neurophysiological stimulation with agitation on behavior. For that, we are also implementing, we are starting a project with ITT data we have the tertiary unit for autism with comorbidity, very severe children under 18, that they have not responded to any treatment. 95% of them, they have very severe conduct disorder, aggression, autolysis, and now we are going to implement that they carry a risk for electrophysiological measures because they have problems for communication, but also to recognize emotional cognition, and we are developing, we have cameras in our unit, and we are developing how we are going to relate the high stimulation, neurophysiological stimulation at the low, with all our cameras and registration. Apart from that, we have an increase, a very important increase of mental disorder in children and adolescents. For example, in the last 3 years, we have a 127 increase of anxiety disorder, 33 of depression, 21.6% of eating disorder increase, and more than 195 of suicidal attempts. These are data from the Catalonia. That has carried on an increase of psychotropic medication, a very important increase, and around between 30 and 50% of children and adolescents, they don't respond properly and develop very severe side effects. The most severe, they are metabolic syndrome, especially obesity, cardiovascular problems, and increase of suicidal ideation. They have more severe side effects, and even they are higher for children with psychotropic medication than adults. Usually, there is the first experience of medication, and if they have side effects, usually the family and the children, they don't want to take medication, and usually the more severe mental health disorder, they start in children and in adolescents. We have a great problem because there is a lack of pharmacological treatment trials under 18, and most of the medication that we use is off-label, and that is a risk for the children. The pharmacogenetic aims is to provide information on mechanism of action of drugs, not the diagnosis, and to identify pharmacogenetic markers to personalized treatments. Not everybody responds equally to pharmacotherapy, and on average, 30 to 50% of people don't respond adequately, and they have side effects. The objective of our pharmacogenetic unit is to develop rapid and low-cost assays for the determination of key pharmacogenetic variants, development of accurate and clinical-friendly recommendations for clinicians based on pharmacogenetic profiles, and to implement protocols of pharmacogenetic interventions and also perform research. We are a unit that we have 50,000 children and adolescents every year, and we have 1,500 kids, 1,500 first visits. We have implemented for every child and adolescent in our service that we start a medication, a pharmacogenetic testing. And we have, since 2021, we have more than 800 patients that we have implemented pharmacogenetic testing. Now we have several grants that you will see that we are doing with all the psychiatric disorders. Usually, we have the results of the pharmacogenetic testing in 2 to 5 days, and it's easy to implement the results in our decisions. Depression is associated with decrease of serotonin and noradrenaline neurotransmission, and the SRIs activates the serotonin pathways by blocking the serotonin transporter and the reuptake of serotonin in the synapses. For that reason, we perform analysis especially for the variants of the genes implicated in the action of the drugs, but also in the proteins that metabolize the drugs. We have seen that we have very high variance, especially of how the variants, the gene variants, have an influence in the metabolizing of the drugs. And we have, for example, in our example, that a very high number of ultra-rapid metabolizers that we have to duplicate or triplicate the doses of drugs. But also we have a high number of poor metabolizers that we have to adjust the amount of medication in order not to have side effects. Also, the antipsychotics are used in children and adolescents. We have the first psychotic experiences, and also we have a very high variance of the CYP metabolizing enzymes, and also we have in the antipsychotics a very high variance of ultra-rapid and intermediate and low metabolizing. Also, we are doing studies, also there are not a lot of research in pharmacological treatment for ADHD. This is the results that we have for one of the examples that we have with 42 adolescents and adults with autism, with a case control study, that we found that all of them, they have problems about the target where the antidepressants work, and also with the metabolizing. When we implemented the pharmacological testing, we have a good improvement in the CGI, in the clinical global impression, in 38 out of the 41 examples of the autistic patients. Even 22 of them, they stabilize with a comorbidity, not with autism. Yeah, I'm finished now. And also with the GGAS, that is also the Children Global Assessment, we have an improvement in 37 out of the 41. We use Salive, it's very easy. All the team is trained in how to use the pharmacological testing and the pharmacogenetic. This is a result, just to finish, of a 13 adolescent that I know him since he is 3. This has a mild mental retardation, but he's very functional. And he had a very severe psychotic disorder that he was in hospital, very aggressive. He was restricted. He was in another hospital. In our unit is forbidden any kind of physical restriction. And then he came to our unit, and this is the result of the test. With most of the drugs, he didn't respond properly, and we have to increase to clozapine 3 times the amount of medication, and he improved very well. In fact, 2 years after, he is very well. And just to finish, that are the grants that we are having and the publication.
Okay, thank you very much, Amaia Hervas, doctor from this Hospital Universitario Mútua de Terrassa. Now, group number 3 with MyMask, development of a 3-dimensional individualized mask for neonatal non-invasive ventilation with Nuria, Massip, and Berta, Palomer, industrial design engineering from Elizabeth School, collaborators in neonatology service in Hospital Clínic of Barcelona. Non-invasive masks are designed based on generic facial models, therefore difficulties in fitting patients. Unique characteristics are common. A poor fit of the mask may have consequences such as air leaks or pressure ulcers. It's possible to optimize, isn't it, the fit of interfaces by adapting them to a patient's face. So here you are, the stage is yours, 10 minutes. Okay, thank you very much. MyMask with Nuria and Berta.
Hello, good morning, everyone. We are truly excited to be here today and to share this, to have the opportunity to share this project. Well, first of all, we would like to present ourselves. My name is Nuria Massip, and she's my colleague, Berta Palomer. We are here representing Óscar García Algar from Neonatology Unit at the Hospital Clínic Barcelona. So our project is based on customized nasal mask for neonatal non-invasive ventilation. It is important to understand that a premature newborn is an immature patient who requires respiratory support during the first days of life. So, non-invasive ventilation through nasal interface in neonates is a crucial intervention developed to improve respiratory outcomes, reducing invasive procedures. So, NIV is a positive pressure. Through an interface, positive pressure is applied to the airway, maintaining its patency. NIV is performed in both full-term and preterm newborns, and currently there are only 4 different sizes of commercial interfaces, which are not adaptable and do not fit properly to the facial features of the patient. So let's move on. Who are the candidates of our solution? So on the one hand, we are applying NIV to a 10% of all term newborns who receive resuscitation or stabilization in the delivery room, to 80% of newborns who need respiratory support. But who are the candidates for our solution? The ones we are focused on are the 75% of premature newborns and the 20% of newborns in the NICU who they both receive respiratory support. So, prematurity is a global problem with a rate of between 7 and 13% of newborns. Also, improper fitting due to the low efficiency of commercial masks can lead to several problems, such as pressure ulcers, air leaks, or cranial deformations due to fixation. So let's move on to the diagram below. As we can see, approximately 100 of critically premature newborns are born in our hospital each year. Over 70% of them will require non-invasive ventilation for average 8 days. And from these, 30.7% will develop pressure ulcers during the first week of life. As we can see, the problematics are highlighted on the images. Our solution is the design of individualized and adaptable nasal masks for non-invasive ventilation destined to newborns using 3-dimensional scanning and bioprinting. As we can see, we have divided the process into 4 stages: scanning, modeling process, additive manufacturing, and clinical use. The first phase of our process is technology, the scanning. The 3D scanning of the newborn, placed in a supine position, was carried out using 3D Peel 2 CAT scanner. It is worth to mention that the scanner is done up to a certain distance to avoid disturbing the patient. As we can see in the images, first we have done the scanning, then how we capture the image, and finally the result of the morphology 3D model. The second phase of the process is the design. Modeling process, image processing. So for this, we use Mimic's Medical 24 program from Materialize to customize a mask that perfectly fits the geometry of the newborn with a smooth contact boundaries, as we can see on the upper images. Then, the third phase is the 3D printing. The created design was sent for printing in an STL format. The masks were printed with biocompatible silicone, but now we are working on to the material selection.
Let's continue with the phase 4 of our process. As we can see, it's the clinical use. It is important to know that the personalized mask fit perfectly the features of the newborn, reducing the air leaks and avoiding overly the forced fixation. Under the same conditions and with a constant airflow, commercial masks exhibited more than 75% of air leakage, whereas this value was severely reduced with our individualized masks. As we can see in the image below, our project is the blue one and our commercial mask is the green one. As we can see, the pressure is uniformly distributed all over the nose of the newborn, whereas in the commercial masks has different marked points. MyMask is the name of our project, and we think that it's a market disruption. What we have done is a comparison between our 2 competitors, our 2 most important competitors. On the first hand, we have Fichet and Peckel, and on the other hand, we have Drager. As you can see in the diagram, our project provides less pressure ulcers, less air leaks, less cost, less deformations, and taking into account all this, we can say that we are more efficient. Let's talk about the degree of disruption in our future activities. It is important to know that some tests have been done with a preterm mannequin, and this gives us some results about the reduction of air leaks, and a non-aggressive capture from the image was done, as well as a certified silicon. Nowadays, we can place ourselves in a TRL between 5 and 6, and in a IML between 2 and 3. Talking about the clinical trial, it is important to know that it was done with mannequins, and these results told us that our algorithm worked, as well as we have to say that some compassionate cases were done thanks to the collaboration with the Hospital 12 de Octubre. Our current situation and where we are nowadays, we can say that we are working on the IAMS permission, as well as we are collaborating with the Hospital 12 de Octubre and the Universitat de Barcelona for the 3D printer. We are as well validating different materials such as silicone and resin for medical use. And we are pending for the manufacturer accreditation and testing a very important shear stress absorbent cream that is placed between the face of the newborn and the mask. 3 MyMask, our benefits and our impact. We can say that our project is a process that can be applied in a wide variety of sectors. So we can say that we are scalable, we have scalability. It is adaptable to the features of each newborn, so we can say that we are adaptable. We have different collaborations, as well as we are biocompatible, using a biocompatible material to prevent potential irritations. We can say as well that one single mask from 3 MyMask is more cost-effective than a pack of 4 commercial ones, so we can say that we are costless. Through different compassionate cases done thanks to the collaboration with the Hospital 12 de Octubre, efficiency have been demonstrated, so we can say that we are efficient. And last but not least, Hospital Clínic de Barcelona counts with a clinical expertise and a clinical professionalism that can help to lower the less side effects of the masks. For finishing, we can say that 3 MyMask counts with 2 different publications. The first one is from the Hospital Clínic de Barcelona, where the clinical trial with mannequins is perfectly explained. And the second one is a scientific publication from the Hospital 12 de Octubre, where the different compassionate cases are explained. Our project counts with a very good group. We are formed by the Hospital Clínic of Barcelona with Cristina, Mario, and Oscar, as well as all the other collaborations and us that we are from University of Elisava. Thank you very much for your attention.
Amazing, Nuria and Berta. Well done it. And you have one minute more. So thank you very much for this brief approach to this MyMask. So we need the last but not the least, group number 4, artificial placenta, with Adrià, Escolà, Innovation and Business Development Manager in the Barcelona Natal Fetal Medicine Research Center. This is a MSc in Biomedical Engineering. Adrià has worked as a project manager of the artificial placenta and has experience in venture capital investment in cash capital risk, startup acceleration and corporate open innovation in Chip2B Foundation. And before joining this artificial placenta project at BC and Natal, Adrià led also company creation and portfolio management activities at Vall d'Hebron Institute of Research. So well done it. So Adrià, Escolà, it's your turn. Big applause for him. Last but not the least, this artificial placenta. Here we go.
Now? Yeah. Thank you very much. Well, today I'm going to present the Caixa Research Artificial Placenta project. It's an experimental project that we are performing at Barcelona Natal. First of all, let me introduce the problem. The previous speakers already introduced the prematurity, but as you may know, the majority of pregnancies usually last for 9 months. This is between 37 and 41 weeks of gestation. And there are around 10% of the people that is born before. We are focusing in the people that is born around 6 months. This is the extremely preterm or extremely prematurity. And it's a significant amount of people with huge problems. As you can see here, they have a high rate of mortality and also a high rates of major sequels. Almost all of the extreme prematures are going to face major complications later in life. And this problem already affects 35,000 newborns per year in Europe. It may not seem a huge number, but if you compare it with other illnesses that we are more aware of, it is a great number. For example, if we compare it with childhood leukemia, it affects around 6,000 kids per year, and almost all of them survive the treatment without complications later in life. So these extremely preterm babies have a worst scenario to face. And this is why we usually don't know about this problem. It's because almost all these extremely preterm children are going to face problems like vision loss, brain damage, hearing problems, heart diseases, and not all of them are going to suffer the same problems or even all of them. So this is why we are calling this problem the invisible multisystemic problem. And maybe you're thinking, okay, but the prematurity rate for sure is decreasing among the last years. But the truth is the data showing that there is an increase on the preterm birth incidence in Europe, but also in worldwide. And this is because there are a lot of factors affecting the increase of this incidence, like the age of pregnancy, the lifestyle, the assisted reproduction, or maybe the short interpregnancy. So what we can do is to treat these patients in a way that we could reduce the sequels or the major complications they're going to face later in life. So nowadays, we are treating them as good as we can by intubating them, by feeding them by the nose, by supplying them oxygen. But the reality here is that there is a biological barrier because the lungs of this fetus, even if we put a lot of oxygen, their lungs are not still developed, are not mature, so they cannot breathe. And sometimes this treatment also increases the sequels they are going to face later in life. So we are proposing a change of paradigm. We're proposing to treat these babies as a fetus, not as a newborn, because they are not able to survive as a newborn yet. And we are reproducing what happens in the majority of pregnancies. If you think what is the natural transcourse, the fetus are inside a cavity, the maternal womb, that protects the fetus from noises, from everything. It's a safety place where a placenta, that's an external organ, provides oxygen and also nutrients for the fetus to keep growing and keep maturing their organs, so it could be able to survive outside the mother's womb. What we are proposing is to recreate this womb and the placenta, but artificially outside the body of the mother. So we have here the chamber where the fetus is going to grow in a safety measures, and also this artificial placenta that is supplying oxygen and nutrients and also medicines to the fetus, only connected by the umbilical vessels. And the blood is pumped by the fetal heart itself. So here is what we want to accomplish, to reduce the major sequels, major complications, and also the mortality by a huge deal. And just to make an example, if we take a 24 weeks of gestation fetus, and we can maintain it during a month in our solution, we should expect a reduction from 30% to 10% of mortality, and also a reduction of major complications from 80% to 30%. So, why we are doing this in Barcelona, or why we are developing this project here in BCNatal? Well, because BCNatal is a large fetal and neonatal clinical research center based in Barcelona. It integrates two first-class university hospitals, like Hospital Clínic de Barcelona is one of the main hospitals in Southern Europe, and also Hospital Sant Joan de Déu, that is one of the top-tier hospitals specialized in children's. And also because our team, led by Dr. Eduard Gratacós, has a large experience in fetal surgery. In fact, he was the one who introduced this technology into Spain when it was only an experimental therapy in a few centers in Europe and the States. And nowadays, we are ranked top 10 as a fetal surgery center and performed more than 2,000 cases already. And all of this is happening with this project. We started in 2019. Thanks to a funding from La Caixa Foundation, we could start animal experimentation in 2021. And we are keeping improving our prototype and our system. And by November 2023, we achieved 14 days of survival in our system with lambs. This is happening because all of this team, we are more of 50 people, highly qualified from physicians to engineers, and also a select group of collaborators that are helping us to keep improving the system and our technology. And this is the system. As you can see, the protected environment is reproducing the mother's womb. And also the extracorporeal circulation circuit and the nutrition is going through this artificial placenta that connects only through the umbilical vessels. And for us, it's really important also the monitoring system, because we are not touching the fetus. The fetus is safe inside this chamber. So we need to monitorize continuously the fetus by the outside. And we have this system that allows us to have information 24/7 continuously from tests, from the fetal health conditions, actions performed to the fetus, and also sensors from the system. And so far, there are 5 research groups around the world performing animal models like us. We were the last ones to join this select group, and yet we are already toe-to-toe with the better ones. But for us, it's not a race to be the first one, but a path to demonstrate that these systems will have a huge impact on extremely preterm babies. So thank you very much, and I hope you enjoyed the presentation.
Perfect. Thank you very much to Adrià, Escolà, with this so important artificial placenta. Keep improving like this with this innovation and business development group team. Thank you very much. At the end of this morning,